Welcome back to your 5th Essential Genetics video. This video is on transcription. But what is transcription? Good question, my lady. It's the first stage in a process called protein synthesis. But what's protein synthesis? Protein synthesis is the process of making proteins. This process is also called gene expression, for a reason I'll explain later. But what is a protein and why do we care? A protein's like DNA, it's a polymer. It's made of building block molecules, smaller building block molecules. In DNA, the building block molecules are DNA nucleotides, and there's four types of different building blocks you can have in a DNA molecule. In a protein, you could have 23 different types of building block molecules, and they're called amino acids. Fun fact, the human body can make 21 amino acids that we require, but the other two we have to get from plants or animals that have eaten the plants. And proteins do a variety of things for the cell. They're like the henchmen of the cell. They can carry things around the cell. They can act as barriers to parts of the cell. Mainly in the cell membrane, they can let certain substances in and out. They can be structural support for the cell. They can build these great long structures that can give a cell resistance to compression forces or stretching forces or whatever, for example, and a bunch more functions that I haven't explained. And they can be a certain type of protein called an enzyme, which is what we call biological catalyst. It starts a chemical reaction or speeds a chemical reaction up. If you think about it, if you want to digest a piece of sugar, on its own, if you just leave that in the air with like no living things in it, it'll just take ages to decompose or to be digested. If you put enzymes on that, that'll happen quickly. However, proteins have a very specific structure to meet specific function. There's loads of different types of protein. You need a very specific one to digest, lactose, for example. Lactase. That's an enzyme that digests lactose. It needs a very specific structure, it needs a very specific amino acid composition. How is that determined? How can the cell recreate lactase over and over and over again, considering there's so many different possibilities, so many different combinations of amino acids you can make, and so therefore so many different fucking proteins, pal. And you probably have a hunch about this already, but DNA determines the structure of the protein you're interested in. So let's say it's lactase. There will be a lactase gene, a piece of DNA somewhere in the cell that the cell will use to determine the structure of lactase, like some sort of script, like it's some sort of instructions that it uses to recreate lactase the same way every time. This is useful because we always want to digest sugar, right? Or maybe more important processes like, brain shit. It seems pretty important. But a piece of DNA that's used as a script for a protein structure, it's called a gene. So over these next couple of videos, I will describe to you the exact process of how we get from the information contained in the gene on how to build the protein, or what amino acids to include in the process, to the finished product, the finished protein itself. Protein synthesis is quite different in prokaryotes and eukaryotes. Prokaryotes are just cells without a nucleus around their DNA. It just sort of floats around in the cell. Eukaryotes have a sort of a bubble of like fat around the chromosomes called a nucleus. And a chromosome is just structures DNA is organized as, which we might cover in another video. We'll see. Surprisingly, despite being called essential genetics, it's not really required in this series, but I might just cover it anyway, because I'm a bad boy. I've drawn a prokaryote there and a eukaryote there. Now, in transcription, a copy of the gene is made, and this copy of the gene is then brought to a part of the cell where it's used to determine, as I've said many times in this video, the structure of the protein. In prokaryotes, it just goes strictly to this stage, directly. It goes from transcribing the gene, making that copy of the gene, straight to using it to determine the structure of the protein. So, transcription. And the stage of using it to determine the protein structure is called translation. This is in prokaryotes. In eukaryotes, it's not the same. There's 2 extra stages that it has to go through. After you've done your transcription, you must modify your copy of the gene. So that's called post-transcriptional modification. Then it's translation. I'll shorten this here so I can be quicker. And then translation. But even though the protein's made, we might still need to modify it even more. So post-translational modifications, shortened in again there. Pretty much in transcription, what happens is, I said that a copy of the gene. It's not just any copy of the gene, it's an RNA copy of the genes that's made. But you might be wondering, what the hell's RNA? Well, the difference between DNA and RNA is that the sugar in RNA is ribose, and we see that, of course, an RNA molecule is a polymer like a DNA molecule. Instead of being DNA nucleotides as building blocks, it's RNA nucleotides in RNA. And of course, the DNA nucleotide building blocks vary in the nitrogenous bases that they have. Both DNA and RNA nucleotides can have cytosine. So I'll just shorten cytosine down to C. You can both have guanine. You can both have adenine. However, exclusive to DNA is thymine, and exclusive to RNA is uracil. Now, uracil and thymine pretty much operate as if they're the same molecule, or nitrogenous base. They're both complementary to adenine. And if you have no idea what I'm on about, you should probably check out the first video in this series on DNA structure. Anyway, RNA, the transcription can be broken down into 3 steps: initiation, elongation, and termination. Let's go into initiation now. Let's begin with kids screaming. So we've got our DNA molecule here that has the gene that we're interested in on it. So let's draw that really quickly. Now, if you know how DNA replication goes, this is not like it. There's a lot of similarities, but they're not the same process, obviously. This enzyme called, we'll say this is the enzyme, called RNA polymerase, attaches to a site or a DNA sequence on the DNA molecule called a promoter. Now, in prokaryotes, the RNA polymerase can just attach to the promoter just like that. In eukaryotes, RNA polymerase needs help from a whole group of proteins called transcription factors. Transcription factors are just proteins that help out or have some sort of role in transcription. There's loads of these motherfuckers, right? I'm not going to name them all because I don't know them all, quite frankly. Yeah, so with the help of transcription factors in eukaryotes, RNA polymerase will bind to the promoter. So I'll just say that's the promoter. I can't spell all that good, forgive me. And RNA polymerase binds to, either with the help of transcript, again, I'm just repeating myself at that point. When it does so, in DNA replication, the hydrogen bonds between the DNA strands were broken by DNA helicase. This is not the case over here. RNA polymerase, and RNA polymerase can just do this by itself. Break the hydrogen bonds between the DNA strands so they get separated out. Again, I've just drawn the process out for you here. This is RNA polymerase, and it's going, now these directions are relative to the strand that it's on. It's going towards the 5' end of the DNA molecule. Because then it can add RNA nucleotides to the 3' end of the RNA molecule that will eventually make in the next stage. Topoisomerase is here though from DNA replication. If you don't know what happens, as you unwind the DNA molecule, you break the hydrogen bonds between it. You generate a lot of stress just ahead of this bubble here, which in this case is called a transcription bubble. You don't want to generate so much stress that you break the molecule. So what topoisomerase does is it temporarily breaks the molecule and reattaches it so the stress from the unwinding from the RNA polymerase is reduced. If you want more information on that, check out my second video in the series on DNA replication. So in the next stage here on elongation, RNA polymerase just adds RNA nucleotides to the growing RNA strand. And it doesn't need a primer. Like in DNA replication, doesn't need that. Nah. Yeah. Just forget it. You're not saying anything, Bob. I forgot to mention that the white strand that you see here is the RNA. The black strand is the DNA. Transcription only occurs on one strand, which I believe is called on the DNA strand that occurs on is called the antisense strand, for some reason. Anyway, for the final step in termination, RNA polymerase will reach a certain sequence in the DNA molecule called a termination sequence. When it reaches this, it'll just stop what it's doing and it'll detach from the DNA molecule, and then voila, you've got yourself an RNA molecule. Specifically, what's called an mRNA molecule, which stands for messenger RNA. In prokaryotes, this is the end of the story. It would just go straight to the translation stage. In eukaryotes, it still needs modified. So this version of the mRNA molecule is called the primary transcript, or pre-mRNA. We'll cover that next video, but we're not done quite yet. We need to go over a bit of regulation in eukaryotes. I didn't know where to place this, to be honest with you, so I'll just chuck it in this video. Anyway, how eukaryotes regulate the rate of transcription, so how often the gene is turned from, how often, sorry, an RNA copy of that gene is made. To do it in the following. There's a special sequence that can be located far away from the gene, the promoter site in the gene, right next to the promoter. If we're talking about an enhancer sequence, a specific type of transcription factor called activators will bind to the enhancer sequence, and with the help of other proteins, will bend the enhancer sequence to near the promoter. So I've just drawn that here. So the enhancer sequence, the enhancer DNA sequence, is being bent over to the promoter. Let's just say the enhancer sequence is far away. And what happens is the activators interact with the promoter sequence in such a way that it increases the rate at which the gene that the promoter sequence is for is turned into an RNA copy. We can have the opposite effect by decreasing the rate of transcription, but this would be the opposite of an enhancer, and that would be called a silencer. And the transcription factors that would do that would be called repressors, the transcription factors that would move over to the promoter region. Anyway, that's us. Hope you enjoyed. Next video we'll be talking about post-transcriptional modification. Hopefully I'm better. Right, bye-bye. Oh hey, the slippers have returned. Don't forget to check the description-friendly specifications, because there are a couple of things in this video that you might get the wrong impression of if you're just listening to my coronavirus voice from the video. Right, bye.